concerning the last paper in our session, which will be presented by Dr. Dino Samartzis. Title is A Novel Pathway Associated with Lumbar Disc Degeneration, a Genome-Wide Association Study.
Thank you. Thank you very much for the opportunity to present our work here today. So as we all know, disc degeneration is just one of the factors that's associated with low back pain. However, there are numerous determinants related to the development of disc degeneration. One very important one is genetics. And approximately 20 years ago, we had a very seminal study published by Michel Batte and Tapio Wiedemann, based on a twin study noting that lumbar disc degeneration has quite a bit of a high heritability rate. Well, ever since then, numerous investigators worldwide have attempted to identify various genes related to lumbar disc degeneration, and in large part this has been based on a candidate gene approach, but there are numerous limitations and biases with this, especially that this is based on the fact that we are using priori knowledge to identify your specific gene, and many of these genes have not been replicated. So one of the more preferred methods is to conduct a genome-wide association study. And the first one that was conducted, and the only one to date, was reported by Francis Williams, who's here with us today, looking into a Caucasian group of Northern European cohorts, mainly composed of MRI, CT scans, and X-ray scans. Just to give you a little bit of background, a genome-wide association study, or GWAS design, basically you have your cases, which you identify, those who have some degree of lumbar disc degeneration, and those who are controls, usually normal individuals. You obtain blood or saliva, you put them through some type of chip, you analyze their DNA to identify certain SNPs, or single nucleotide polymorphisms, which generally are high-resolution markers for gene mapping of a certain condition. However, there's certain challenges with regards to GWAS and lumbar disc degeneration. I mean, disc degeneration is a pretty common condition, but the phenotyping is quite complex, and this thing needs to be standardized. But one way we've kind of gone around it is we've actually identified two different types of lumbar disc degeneration. This is a study here, a paper, noting our thoughts about this element of a developmental component of disc degeneration, or disc generation in a sense, and from this stem, this study that we presented here last year, where we identified two distinct origins of disc changes, where we divide the upper regions to the lower regions, that are ones that are not age-related, others that are age-related, and we actually developed an interesting scoring system based on this. So with this in mind, the objectives of this study were to, utilizing a new etiological classification scheme of lumbar disc degeneration on MRI, we addressed a genome-wide association study of disc degeneration in an Asian population. This was based on our Hong Kong disc degeneration cohort study, of which to date we've recruited over 3,500 Southern Chinese volunteers, of which all of them we've obtained MRIs. And we basically used this two types of scoring methods, looking at the upper lumbar score and the lower lumbar score based on MRIs. We've genotyped approximately 2,373 individuals from the cohort, looking at over 800,000 SNPs. We utilize this Illumina Omni Xianghua B chip, which is very good for this population. We address single variant association analyses as well as pathway-based association tests to somehow increase the power. With regards to quality control, as I mentioned, we had over approximately 2,400 samples that were genotyped, but after weeding out people that were related to each other, or bad genotyping, etc., we ended up analyzing 2,116 subjects for GWAS of lumbar disc degeneration. With regards to our results, when we conducted principal component analysis looking at the population structure, we saw that the Chinese population was a bit distinct and had a bit of a distance with other northern Chinese and Japanese populations. Based on the variant-based association studies, looking at if there was any hits or SNPs of suggesting something, we didn't see any significance. But when we looked at pathway-based association tests, and this was mainly used to boost up the power of the study, basically looking over 1,300 different pathways based on this particular reservoir here, we saw that the upper lumbar score was not significant at all, but we saw something very interesting here when we utilized the lower lumbar score of disc degeneration. We saw some type of moderate form of significance, and in particular this interesting pathway here. So after we corrected for multiple testing, in fact, we saw that there were 9 genes that reached some type of statistical significance, and these were involved with the integrin pathway. Now, for reasons because this is not published yet and we still need to replicate a lot of these studies, I'm not going to get into what type of genes these are specifically. But in general, integrins are transmembrane receptors that are bridges for cell-to-cell and cell extracellular matrix interactions. And in conclusion, this is the largest genome-wide association study of lumbar disc degeneration on MRI, and the first that I know of in an Asian cohort. We believe the pathway-based association analysis is a viable approach, and that the endocrine pathway is something of interest, but we need to further replicate these studies and collaborate with other ethnic backgrounds to validate what we found. And in conclusion, I'd like to thank all of our collaborators. There were quite a bit of people involved with this, in particular Yandy, who did the statistical genetics. Thank you very much.
Okay, I see people already coming to the mic, so Saki, please.
I am Hasuzume from